Outmaneuver the competition with proactive clinical development intelligence. Discover how. Log in. Drugs. Type your search. Nav. Search section headers. Identification. Pharmacology. Interactions. Products. Categories. Chemical identifiers. References. Clinical trials. Pharmacoeconomics. Properties. Spectra. Targets 7. Carriers 1. Transporters 5. The next evolution of DrugBank is here. DrugBank's latest evolution delivers AI-powered, scientifically validated to accelerate drug discovery like never before. Get started. Explore the full scope of our drug knowledge tailored for pharmaceutical research needs in our data library. Learn more. Amisulpride. Description. A drug used to treat schizophrenia and prevent nausea and vomiting after surgical procedures. Structure. DrugBank ID. DB06288. Modality. Small molecule. US approved. Yes. Other approved. Yes. Patents. 8. Indicated conditions. 4. Clinical trials. Phase 0. 1. Phase 1. 14. Phase 2. 7. Phase 3. 14. Phase 4. 23. Therapeutic categories. Dopamine D2 receptor antagonists. Mechanism of action. 5-hydroxytryptamine receptor 7 antagonist. 5-hydroxytryptamine receptor 2A antagonist. D2 dopamine receptor antagonist. Plus one more target. Identification. Summary. Amisulpride is a dopamine D2 receptor antagonist used in the treatment of acute and chronic schizophrenia, and in the prevention and treatment of postoperative nausea and vomiting in adults. Brand names. Barhemsys. Generic name. Amisulpride. DrugBank accession number. DB06288. Background. Amisulpride is a benzamide derivative and a dopamine receptor antagonist that selectively works on dopamine D2 and D3 receptors. As an antipsychotic agent, amisulpride alleviates both positive and negative symptoms of schizophrenia, and it exhibits antidepressant properties in patients with psychiatric disorders, dysthymia, and major depression. 5 Amisulpride predominantly works in the limbic system, which explains its relatively lower risk of extrapyramidal adverse effects compared to other atypical antipsychotic agents. 2. 11 oral tablets of amisulpride is used in European countries as a treatment for acute and chronic schizophrenic disorders, as well as secondary negative symptoms in mental health disorders such as affective disorders, depressive mood, and mental retardation. 11. Amisulpride is also used as an antiemetic agent. In the US, the intravenous formulation of amisulpride is used to treat and prevent postoperative nausea and vomiting in adults, either as monotherapy or in combination with another antiemetic agent of a different drug class. 10. It is marketed under the brand name Barhemsys. Modality. Small molecule. Groups. Approved. Investigational. Structure. Weight. Average. 369.479. Monoisotopic. 369.172227057. Chemical formula. C17H27N3O4S. Synonyms. Aminosultopride, amisulprida, amisulpride, amisulpridum. External IDs. APD 421. Stay ahead of the competitive shifts that matter. Pharmacology. Indication. Intravenous amisulpride is indicated in adults for the prevention of postoperative nausea and vomiting, either alone or in combination with an antiemetic of a different class. It is also indicated for the treatment of postoperative nausea and vomiting in patients who have received antiemetic prophylaxis with an agent of a different class or have not received prophylaxis. 10. Oral amisulpride is indicated for the treatment of acute and chronic schizophrenic disorders, characterized by positive symptoms with delusions, hallucinations, thought disorders, hostility and suspicious behavior, or primarily negative symptoms deficit syndrome, with blunted affect, emotional and social withdrawal. Amisulpride also controls secondary negative symptoms in productive conditions as well as affective disorders such as depressive mood or retardation. 11. Build, train, and validate predictive machine learning models with structured datasets. Associated conditions. Indication type indication combined product details approval level age group patient characteristics dose form. Treatment of acute schizophrenia 12 characters. 9 characters 6 characters. Treatment of chronic schizophrenia 12 characters. 9 characters 6 characters. Treatment of negative symptom 12 characters. 9 characters 6 characters. Treatment of negative symptom 12 characters. 9 characters 6 characters. Treatment of negative symptom 12 characters. 9 characters 6 characters. Contraindications and black box warnings. Avoid life-threatening adverse drug events with our clinical API. Pharmacodynamics. Amisulpride is a selective dopamine D2 and D3 receptor antagonist with no affinity towards other dopamine receptor subtypes. Amisulpride is an atypical antipsychotic agent that works as an antagonist at dopamine receptors in the limbic system. Since it works preferentially in the limbic system, amisulpride is less likely to be associated with extrapyramidal adverse effects than other atypical antipsychotic agents. Amisulpride has no affinity for serotonin, alpha-adrenergic, H1 histamine, cholinergic, and sigma receptors. In clinical trials, amisulpride improved reduced secondary negative symptoms, affective symptoms, and psychomotor retardation in patients with acute exacerbation of schizophrenia. Notably, amisulpride has a differential target binding profile at different doses. At low doses, amisulpride selectively binds to presynaptic dopamine autoreceptors. At high doses, it preferentially binds to postsynaptic dopamine receptors. 3 This explains how amisulpride reduces negative symptoms at low doses and mediates antipsychotic effects at high doses. 111 study alluded that the antinociceptive effects of amisulpride are mediated through opioid receptor excitation and D2 receptor antagonism. 3. 5 The actions of amisulpride at opioid receptors may explain its pro-convulsant properties. 5. Amisulpride is also an antiemetic agent that prevents and alleviates postoperative nausea and vomiting. It primarily works by blocking dopamine signaling in the chemoreceptor trigger zone, which is a brain area that relays stimuli to the vomiting center. 11. In clinical trials comprising Caucasian and Japanese subjects, amisulpride caused dose and concentration-dependent prolongation of the QT interval. Thus, intravenous infusion under a strict dosing regimen and close monitoring of patients with pre-existing cardiovascular conditions are recommended. 10 Amisulpride increases plasma prolactin levels, leading to an association with benign pituitary tumors such as prolactinoma. 11 Mechanism of action. Dopamine is an essential and critical neurotransmitter produced in the substantia nigra and ventral tegmental regions of the brain. Dopaminergic projection function in the nigrostriatal, mesolimbic, and mesocortical systems. Hyperactive dopamine transmission in the mesolimbic areas, or dopamine dysregulation in various major brain regions, is understood as the key driver of positive and negative symptoms of schizophrenia. 9 Many antipsychotic agents act as D2 receptor antagonists, as with amisulpride. 2 Amisulpride is a selective dopamine D2 and D3 receptor antagonist. 10 It has high preferential activity towards dopamine receptors in the limbic system rather than the striatum, leading to a lower risk of extrapyramidal side effects than other atypical antipsychotic agents. 4. 6 At low doses, amisulpride reduces negative symptoms of schizophrenia by blocking presynaptic dopamine D2 and D3 receptors, increasing the levels of dopamine in the synaptic cleft and facilitating dopaminergic transmission. 4 At higher doses, amisulpride blocks postsynaptic receptors, inhibiting dopaminergic hyperactivity. This explains the drug improving positive symptoms. 1 Amisulpride also works as an antagonist at 5-HT7A receptors 8 and 5-HT2A receptors, 4 which may be related to its antidepressant effects. 5. The chemoreceptor trigger zone, CTZ, also commonly known as the area postrema, AP, is an important brain region located within the dorsal surface of the medulla oblongata. CTZ is involved in emesis. It contains receptors, such as dopamine receptors, that are activated in response to emetic agents in the blood and relay information to the vomiting center, which is responsible for inducing the vomiting reflex. 7 Amisulpride is an antiemetic agent that works to limit signals that promote nausea and vomiting. Amisulpride binds to D2 and D3 receptors in the CTZ, leading to reduced dopaminergic signaling into the vomiting center. 10. Target. Actions. Organism. A5-hydroxytryptamine receptor 7. Antagonist. Humans. A5-hydroxytryptamine receptor 2A. Antagonist. Humans. AD2 dopamine receptor. Antagonist. Humans. AD3 dopamine receptor. Antagonist. Humans. NMU type opioid receptor. Agonist. Humans. N-delta type opioid receptor. Agonist. Humans. N-kappa type opioid receptor. Agonist. Humans. Absorption. Following oral administration, amisulpride is rapidly absorbed with absolute bioavailability of 48%. 1 Amisulpride has 2 absorption peaks, with one rapidly achieved within 1 hour post-dose and a second peak occurring between 3 to 4 hours post-dose. Following oral administration of a 50 mg dose, 2 peak plasma concentrations were 39 plus or minus 3 and 54 plus or minus 4 nanograms ml11. Following intravenous administration, the peak plasma concentration of amisulpride is achieved at the end of the infusion period and the plasma concentration decreases by 50% within approximately 15 minutes. The AUC 0 infinity increases dose proportionally in the dose range from 5 mg to 40 mg, which is about 4 times the maximum recommended dose. In healthy patients receiving intravenous amisulpride, the mean SD Cmax was 200-139 ml at the dose of 5 mg and 451-230 ml at the dose of 10 mg. The AUC ranged from 136 to 154 nanograms by HML in the dose range of 5 mg to 10 mg. In patients undergoing surgery, the mean SD Cmax ranged from 127.62 to 161.58 ml at the dose of 5 mg. At the dose of 10 mg, it was 285-446 ml. The AUC ranged from 204 to 401 nanograms by HML10. Volume of distribution. Following oral administration, the volume of distribution is 5.8 L kg 11. Following intravenous infusion, the mean volume of distribution of amisulpride is estimated to be 127 to 144 L in surgical patients and 171 L in healthy subjects. 0.10. Protein binding. Plasma protein binding ranges from 25% to 30% in the concentration range from 37 to 1850 ml. Amisulpride distributes into erythrocytes. 0.10. Metabolism. Amisulpride undergoes minimal metabolism. 1 and its metabolites in plasma are largely undetectable. 2 identified metabolites, formed by deethylation and oxidation, are pharmacologically inactive. 6 and account for approximately 4% of the dose. 0.11 metabolites remain largely uncharacterized. Metabolism of amisulpride does not involve cytochrome P450 enzymes. 0.10. Route of elimination. Following intravenous administration, about 74% of amisulpride is excreted in urine, where 58% of the recovered dose was excreted as unchanged amisulpride. About 23% of the dose is excreted in feces, with 20% of the excreted dose as unchanged parent drug. Following intravenous administration, about 4 metabolites were identified in urine and feces, accounting for less than 7% of the total dose administered. 0.10. About 22 to 25% of orally administered amisulpride is excreted in urine, mostly as the unchanged parent drug. 0.1. Half-life. Elimination is biphasic. 0.6. The elimination half-life of amisulpride is approximately 12 hours after an oral dose. 0.11. The mean elimination half-life is approximately 4 to 5 hours in both healthy subjects and patients undergoing surgery receiving intravenous amisulpride. 0.10. Clearance. The plasma clearance of amisulpride is 20.6 LH in surgical patients and 24.1 LH in healthy subjects following intravenous administration. Renal clearance was estimated to be 20.5 L/hour, 342 milliliters min, in healthy subjects. 0.10. Adverse effects. Improved decision support and research outcomes with our structured adverse effects data. Toxicity. In mice, oral LD50 is 1024 mg kg, intraperitoneal LD50 is 175 mg kg, and subcutaneous LD50 is 224 mg kg. The lowest published toxic dose (TDLO) following subcutaneous administration is 0.24 mg.